Microbial growth. Remember when we started the chapter, we said that microbial growth is defined as an increase in the number of cells rather than the size. Binary fission is a process in which a cell grows to twice its normal size and divides in half to produce two equally sized daughter cells. So here you see we have one cell. It is going to basically divide into two daughter cells. As you notice, most bacteria elongate and divide. So this cell is going to be elongating, so getting longer, and then it divides. What are the steps to binary fission? Well, the cell replicates its chromosome through DNA replication. So here we see that the cell is replicating its DNA. It's also going to double the amount of structures. From there, the cell has a growth of a new membrane. So the growth of the new membrane allows the cell to elongate and grow. The cell also forms a new cell membrane, and then it divides. From there, the process repeats. So E. coli, we said, divides every 20 minutes. So every 20 minutes, E. coli does this process. With binary fission, any given cell divides to form two cells. Then each of these new cells divides into two, then makes 4, then 4 becomes 8, and 8 becomes 16, and so on. The growth rate is often described as a change in cell number. It's used to describe how fast a culture is growing. A generation is the interval of two cells from one. The generation time, or the doubling time, is the period required for cells in a microbial population to enlarge, divide, and produce two new cells for each one that existed. Before. Doubling times vary depending on the species of microorganism and the growth condition. Doubling time does not change until the nutrients become depleted or toxic metabolic products begin to accumulate. E. coli grows with a doubling time of 20 minutes in a rich laboratory medium at optimal temperature. For this reason, food contaminated with only a few of these E. coli organisms can cause food poisoning if not properly refrigerated and cooked. The result is exponential growth, which results in an explosive increase in the numbers of cells. The growth cycle has 4 different phases. The lag phase. The lag phase is the time it takes for the cell to start growing once inoculated. This is the no-growth period. They are preparing to grow. The cells are adjusting to their environment. So they take in nutrients, they're synthesizing essential components, they're repairing any damage, adjusting to new media and nutrients, and adjusting to new concentration of nutrients. This varies depending on conditions and the nature of the culture. The next phase is the exponential or log phase, and this is when the cells are grown exponentially. The population doubles per unit of time. The rate increases with each new generation. So this is when they're the most metabolically active, but when they're the most sensitive. So remember back in chapter 2 we talked about penicillin and how penicillin is going to stop the bridges from being, connecting the peptidoglycan layers. This is when penicillin will work the best because it's growing. It's the most sensitive then. The increasing number of cells use nutrients and produces waste at an ever-increasing rate. Usually an essential nutrient will run out, so the exponential growth can't continue for long. Eventually, the number of dying cells equals the number of cells being produced, and the size of the population becomes stationary. So we are now in the stationary phase. There's no net increase or decrease in the population. The nutrients are running out or waste is building up. but metabolism and biosynthesis is still occurring. So the number of cells that are living or that are being made is now, or the number of new cells is now equal to the number of dying cells. The death phase is when the number of cells are dying is greater than the number of new cells. So during the death phase, cells of most microbial species are dying exponentially. So if we take a look at this picture again, over time, over the number of bacteria, so we're doing exponential, so we have to use log. Remember, the lag phase, nothing's happening because they're adjusting to their environment, they're getting used to it. Once that occurs, then they begin to grow exponentially. Remember, when they're the most active, the most sensitive. But eventually nutrients are going to begin to run out, waste is going to start to build up, so that we hit a stationary phase, where the number of new cells equals the number of dying cells. And then eventually the death phase. So with the death phase, we see that we have more dying cells than cells that are living, and that ends the death phase. Here ends Chapter 3.